The next speaker is John Coe, who's going to be talking about the Chemistry Prize. John is a professor in the Department of Chemistry and Biochemistry. He's also the director of the Chemistry Biology Interface Program and the associate director of the Delaware Biotechnology Institute. John earned his PhD at Columbia University and then did postdoctoral research at the University of California at Berkeley. He joined UD in 1996. John's research area is chemical biology, and some of his current projects include light-activated gene expression and design of chemical agents to restore function to mutationally impaired nuclear receptors associated with disease. John?
Thanks, Doug. So today I'm going to tell you about the Nobel Prize in Chemistry, and it's on the topic of G-protein coupled receptors. And so if you don't know what that is, hopefully I'll be able to explain that to you by the end of the talk. A lot of my biology friends ask me, wow, this doesn't sound like a real chemistry topic. Because this is really at the heart of a lot of biology. But when we think about what chemists do, we certainly go out and we make molecules, but we're also there to understand how things work at the molecular level, how things are structured, and how these molecular structures relate to their function and how they interact. And this story is a story about molecular structure and molecular function. And it unfolds over about a 40-year period. And it starts out with Robert Lefkowitz, who's a professor at Duke University, and Brian Kobilka, who is at Stanford University. And Brian Kobilka, Dr. Kobilka, was actually worked in Lefkowitz's lab earlier in his career. So there's even this sort of transfer of knowledge between the two. But let's start off about why we're interested in these compounds and how they work. So when we think about having cells and how cells function, that's one thing. But how do cells talk to each other? And when we have multiple cells, it's one thing. When you have an entire organism and how they relate, those things are often controlled by small molecules that are used to communicate across distances within the body. We usually think of these within the endocrine system, and of course there's many different hormonal organs within the body that communicate and secrete hormone signals throughout the body. They can be molecules, whether they're peptides or proteins or even just small synthetic, small molecules that can go through the bloodstream and reach a target cell, which is sort of the classic endocrine model. And even in local tissue, we can have these same sorts of chemical signals that either communicate locally in what's called paracrine communication, or even communicate to regulate itself through what's called an autocrine interaction. These things are all extremely important in terms of regulating how an organism, how all the cells, how many trillion are there, are all functioning and communicating together. We hear a lot about genomics these days. One of the problems with genomics, not that there's anything wrong with it, but one of the problems about trying to regulate organisms through the regulation of genes is that they take a fairly long time scale to interact. If you turn on a gene, which is something we do in my lab all the time, this can take place in the minutes to hours time scale, sometimes even over days, before a response is regulated by initiating transcription of DNA to RNA and then RNA to protein. But if you need to respond quickly to some threat, such a bear or something like this, you need something that functions much faster than that. So on this time scale, if this is where the bear attacks, this is where the bear, whoops. Now I got to go through my cheesy animations one more time. So if this is where the bear attacks, this is where the bear eats. You have no time for transcription. So hormones function quickly, and that's why they're necessary for us to function well. So these kinds of responses, turn out, are regulated by several different classes of receptors. And one of the largest ones is the G-protein coupled receptors. Now, one of the reasons they're important for us is because so much of the pharmaceutical agents that we use today act through G-protein coupled receptors. So this is a list of the top 120 brand name prescription drugs, at least in 2010. This is over, and if you looked at them and asked which ones of these interact with G-protein coupled receptors, almost half of them interact with G-protein coupled receptors. This is over a $40 billion a year business for just the compounds that interact with G-protein coupled receptors. And they're everything from, many of these are drugs you can read if this was high resolution. So they're drugs like Lipitor and they're ProAir. They're things that are antihistamines. They're leukotriene inhibitors. Insulin is one of the compounds that this interacts with. Oxycontin is another one that interacts. So these are, all those drugs, many of those drugs you hear advertised on TV act through this class of proteins called G-protein coupled receptors. Now, I was looking at many of these, and there was beta blockers, for those who have high blood pressure, and there's some compounds that are used for treatment of Alzheimer's and stuff, and I was thinking, yeah, most college students aren't thinking that they're going to have problems with, high blood pressure and these old people's diseases yet, so I thought I should give some more relevant examples. So G-protein coupled receptors also interact with things like the cannabinoid receptor is a G-protein coupled receptor. And morphine also interacts with the G-protein coupled receptor. And cocaine interacts with the G-protein coupled receptor. And even methamphetamine interacts with the G-protein coupled receptor. So these things that have rapid responses on the body are all taking place through G-protein coupled receptors. We now know that there's over 800 of these, 800 different G-protein coupled receptors. And that's just in humans, right? And this is an enormous family. We don't yet know what all of these things interact with yet. Many of them we don't know what we call the ligand, the molecule that turns it on is. But that's one of the challenges for the current generation and that to follow. But back at the time that Lefkowitz got started in the 1960s, we knew extremely little about how this all worked. We didn't even know what these things were. In fact, he wrote a few years later, until quite recently, receptors were defined as the first step in a sequence of reactions leading to an observable physiological or biochemical effect. And so at that time, they weren't even sure what it was. When you had a hormone interacting with a cell, was it just somehow modulating the cell membrane? Was it somehow affecting transcription? Was it affecting RNA or maybe DNA? They didn't know. And Lefkowitz played an important role in figuring this out. So early on, they knew that somehow these hormone molecules would get to a cell, and it was hypothesized that there's something there, some sort of receptor, that must be able to differentiate that molecule from other molecules and be able to elicit some response. So there had to be some way of coupling this together. And here we're drawing it as cartoons. But what we're going to find out is in 40 years, when we get to today, we start to know much more about what this process actually looks like. So early on, what Lefkowitz's work was working on was trying to figure out what was this receptor, and was there actually a real receptor? Was it just something, were these hormones just perturbing the way the cell functioned, or is there actually a specific molecular interaction involved? And so he labeled this what's called ACTH, the adenocorticotropic hormone. He actually made a radioactive version of this so he could follow whether or not it was binding to the cell. And he found out that there was a specific number of binding interactions that took place in the cell, indicating there was something that was grabbing onto it. And also that he could compete that binding interaction off. And so there was a unique and specific interaction that was involved. So that's one of the major contributions he made. But he took it a step further. And ultimately, he did a little chemistry, and he coupled norepinephrine onto agarose and did what many of us know as affinity chromatography. And he was able to isolate for the first time what this receptor was. And it's a protein was the answer, right? So it was actually a protein that was binding to this, and he could prove that for the first time. Now, he went on and did some really nice studies. He showed, for example, that he could use that binding interaction to actually measure plasma levels of this hormone, ACTH. He even showed that it was associated with a particular disease, Cushing's disease, that these patients had lower ACTH. So we could take even a disease state and link it back down to a particular chemical interaction taking place on the surface of cells that was all mediated through this class of proteins. And even in further work, he did work to show that tolerance, the fact that you get less of a response if you keep taking the drug, it's not that your dealer's giving you weaker stuff. It's actually because there's a weaker response that's caused by a change in the molecular interaction that occurs at the surface of the cell. So this story, the story of G-protein coupled receptors is enormous, and there's so many complexities to this. And it's extremely difficult to try to imagine what's going on. So, for example, we know that there's molecules that can bind to the receptor and somehow turn on a response, usually the production of cyclic AMP or cyclic GMP. We also know that there's antagonists, molecules that can bind to these same receptors, and they can block the normal on signal from binding and therefore shut it off. But when people started to look at more and more different compounds, they realized it wasn't a simple on-off switch. They realized that as you turn on, as you add more and more of the hormone, you can get things that turn it on a lot. And you can get other things that turn it on only part way. So there was almost like there's different levels of activation. There's even compounds that are called partial or inverse agonists that actually, as you add them, they sort of cause the thing to go activate production of cyclic AMP to a lower level. It's almost like putting it in reverse. It's almost as if on the cell there's this multiple speed activator that you can use to regulate cellular processes. So throughout, this is a fairly early paper, 1976. He says it's still not understood, however, the mechanism by which binding, in this case to the beta-adrenergic receptor, is translated into adenylate cyclase action, i.e. how the receptor enzyme system is coupled. It is clear that binding to the beta-adrenergic receptor per se does not necessarily lead to enzyme activation, since beta-adrenergic antagonists are able to bind to the receptor sites with very high affinity and yet do not alter basal adenylate cyclase activity. So there was clearly that this is far more complex than simply something binding and turning on, something binding and blocking the action. But how does this happen? What is this coupling that takes place? This is what's been going on for the last 30, 40 years. And enormous steps have been made to try to understand this. And so we have to sort of skip forward to the future, and I'm just going to give you a couple of slides to understand a little bit more. So we were able to identify many of these proteins. And this looks like kind of an archaic slide today, but I distinctly remember, even as late as 1990, 1999, and just across the green, seeing many talks that would present a figure that looked just like this, where these are the single amino acid letter codes, and this is a chain of amino acids that makes up the protein that's a G protein coupled receptor. And they did some wonderful biochemical studies. First of all, they mapped out where the hydrophobic residues were, and they figured out that these hydrophobic residues fell within the membrane of the cell. And so this actually stuck itself within the membrane. And you could do some really clever tricks by sticking a protease on the outside of the cell, and it would cut all the stuff on the outside of the cell. And if you injected it onto the inside of a membrane, it would cut off the stuff at the bottom. So they could figure out a lot about sort of the topology of how this worked, but not necessarily the molecular details. They also figured out that this was coupled to what were called G proteins. And G proteins were the ones that took the binding interaction to the G protein, to the receptor, and relayed that to other downstream effectors through secondary messengers. And so sort of the picture that emerged was that a ligand would come and it would bind to this receptor. The receptor would undergo some sort of conformational change upon binding that would allow G proteins, in this case G alpha, sorry my Greek symbols didn't come out so well, which could then dissociate and say bind to adenylate cyclase, that would then in turn, it was the adenylate cyclase that was generating the cyclic AMP. And all these steps were worked out by an army of scientists working around the world trying to figure this whole complexity out. But people still wanted to know exactly how this worked, because very subtle changes to the structure of this ligand, to this red molecule, the hormone that binds it, could actually make something that was a turn-on, that would turn on the receptor, to something that would turn it off, or something that would activate it to somewhere in between. Oh, and just to mention, some of the things that are downstream are things like pKa, phospholipases, PI3 kinases, and it's the G beta and G gamma subunits that activate ion channels, and things like, for example, PDEs, important for another drug class that you need when you get older, are also activated by these downstream effectors. And I'll point out that the Nobel Prize in 1994 was given for G proteins themselves. So they wanted to know a higher resolution molecular structure. And the way to do high resolution molecular structures, the gold standard, is to do X-ray crystallography, in which you crystallize the protein into a regular array and use diffraction patterns that come from an X-ray source to deconvolute high resolution structures. And if you can get a high enough resolution structure, you can actually see where the specific atoms are and how those atoms relate and affect the structure of the protein. So the challenge is that membrane proteins turn out to be extremely difficult to crystallize, and they're extremely difficult to crystallize still in high resolution. And so Kabilka commented in 2007 that for protein structures to be effective guide for drug discovery, for drug design, it is critically important that to maximize the available detail, as low resolution structures can be ambiguous at best and misleading at worst. Now, at this time, they already had, for example, the bovine rhodopsin structure. So this is a G protein coupled receptor that uses light as its ligand. It actually has a ligand bound that receives the light and translates that to changes in its structure. but it was fairly low resolution. And crystallizing proteins that bind to the membranes are extremely difficult because in the process of trying to coax these molecules to make crystals, they often fall apart because the very forces that hold them into the membrane are the kinds of forces you want to manipulate to try to get them to crystallize. And this was just a study that showed back for example, in 2005, 6,000 new structures were being submitted a year to the protein data bank, and about 12 of them were membrane proteins. That'll give you an idea how difficult it is. So of the 85,000 protein crystal structures we have in the protein data bank, less than 0.5% are actually membrane-bound proteins because they're just so challenging to get crystallized. But the breakthrough in GPCRs came out in 2007 when they finally got a G protein-coupled receptor that bound a small molecule, that bound a hormone to crystallize. And they did some really clever tricks to get this to work. So in this case, for example, the red molecule is actually the G protein-coupled receptor, and the green stuff in between is actually a molecule that people had really great luck crystallizing, T4 lysozyme. And by making a fusion, they could stabilize the protein and make it more easily crystallize it. And better yet, stabilize it so that they could add detergents that mimic the cell membrane without causing the protein to fall apart itself. And we could finally get molecular details. We could finally actually see a drug molecule bound into the GPCR and look specifically at the atoms that interacted with it. And we said, wow, that's really great. But it didn't tell us how this thing changed. It tells us what it looks like in a static state. But what did it look like before the molecule bound? What's it look like if there's no molecule bound? And how on earth do these interactions affect the G protein that's on the other inside the cell, connected to the membrane? So they continued on this work, and they eventually got a beautiful structure that started to show two different states of a protein. So in this case, they used an antibody. And I should say one of the really brilliant things that Kabilka did was apply multiple approaches to getting this done. And so in this case, they used an antibody to stabilize it in the antagonist form. And we can see that between the gold structure and the gray structure, there are these molecular changes taking place to the receptor that ultimately are going to relay information to the G proteins that are on the other side of the membrane. And then finally, in just 2011, they got the whole story. So here, for example, here's this T4 lysozyme that was used to help stabilize the structure. And down here is the entire G protein complex with a G alpha subunit, and a G beta, and a G gamma subunit. And now we can see that here's where the hormone binds, and here are those helices that run through the membrane. And somehow when this thing binds, there's changes to those helices that relay information down to the bottom. And not only did they get the crystal structure of this whole enormous complex, but they went a step further to get it both in its active and inactive forms. And so we could now look and say, in this case, we're looking at a green versus blue structure, where the green is the on state and the blue is the off state. And we can actually see what molecular changes occur that relay back to the G proteins. And so here in this case, in green, we see the gold structure, it's all splayed out this way, is in the inactive form. And then when it's activated, there's a large conformational change that takes place in the G alpha subunit that is relayed only by the molecular interactions to these helices that are reaching down through the membrane. So what was at times not even known to be a protein, what was hypothesized that there must be something there interacting with these receptors, has been taken by these two gentlemen to be identified as a protein, to understand that it's actually a one-to-one interaction with its ligand that affects cellular signaling inside. And we now have a complete, or almost complete, molecular description about how that process takes place. And with those high-resolution structures, we now see how this whole story goes, from hormone binding to receptor, receptor having a conformational change that interacts with G proteins. There are structures of how G proteins interact with adenylate cyclases and cause this activation to take place. And with those high-resolution structures, we can also understand why some molecules are able to turn the receptor on, Some molecules are able to turn the receptor on more or less, and some are able to shut the receptor down. And how those, at the atomic details, allow us then to make better drugs with less side effects, with more potency, potentially even avoiding tolerance. So these are all things that we're trying to use from these structures. But it's an extremely complicated machine that nature has created. In fact, it's created 800 of these. And now we have finally the molecular details to see this machine in action. And that's why Robert Lefkowitz and Brian Kobilka deserve the Nobel Prize in chemistry.
Thank you, John. Are there any questions? John, you said that there was a molecular scale explanation for development of tolerance, a reduced response to molecules that might interact with these proteins. Is that a due to a direct interaction with the protein, or is that more complicated?
No, it's more complicated. So there's other proteins that actually interact with the G protein coupled receptors, and there's a switching of partners that can also take place. And those protein partners can be modulated by phosphorylation, for example, to regulate their interactions. So it's dauntingly complex at one level, because it's actually sort of a combinatorial interaction that leads to these responses. But at the same time, it's wonderfully complex, because it's the kind of system that you can see where the body can attenuate and adapt under different conditions to provide a complex array of responses, even using just a limited tool set. So, for example, there's 800 different G protein receptors, but I think there's about 80 or so G proteins. And by using these in combination, instead of making one-to-one, one G protein for one receptor, by using these in combinations, that's how we can create this complexity of responses that takes place in different cells and different cell types.
Okay. Any other questions? Yes, sir.
What they did allowed them to beautifully describe one system. I presume the techniques are also now being applied on all sorts of other systems. Is that correct?
That's correct. So once you get, now it doesn't mean it'll work definitely, right? But the wonderful thing about this is that all of those 800 proteins share the same overall protein architecture. They all have this sort of 7 transmembranes, helices running through. And so hopefully these techniques can be applied elsewhere. But not all G proteins can be expressed very well. Not all G proteins, not all GPCR and G protein coupled receptors can be expressed very well. And there's lots of hard work that needs to be done. But it's an amazing tour de force for them to have been able to pull this off. And there's so many subtleties and experimental tricks that needed to be done to pull this off. It's the initial step. Just if I can give you an idea that when I work in a related field on soluble hormone receptors called nuclear hormone receptors, and once they figured out how to get the first 1 or 2 done, now they have pretty much the whole family crystallized out. So they got 1 done, and within 10 years they had hundreds of these done. So there is a lot to be said that once you figure out the trick for one, you may be able to apply it on. Some people work a decade to get some of those done, but there is some translation of information to other ones. So it's not going to take another 40 years to get the next one for sure.
All right. Thank you very much, John.